Hey everybody, Mr. Scott coming at you. So yesterday we worked on and we observed our pea plants. We observed the traits that they have. So the traits that these two pea plants displayed that were different from one another were the height, so how tall they are, and the flower color. You can see this one has white flowers and this one has purple flowers. So today we're going to be using Punnett squares to look at how we can accurately predict the percentage that a pea plant or basically anything will have a recessive or dominant gene, meaning they will display that phenotype, which is the physical characteristics that we can observe. So each feature is controlled by a gene. The two genes are on different chromosomes. You have hundreds of thousands of millions of chromosomes within your body. So the genes are almost identical, but have different alleles. The alleles for the stem length are represented with lowercase l's, where the alleles for the flower color are represented by F. And it's asking, why do we think there's a lowercase and an uppercase? So if we look at this, the lowercase l's show that these are recessive traits. So lower cases are recessive, and our upper cases are dominant. So dominant and recessive traits. So not all alleles have the same influence. Dominant alleles are more influential, whereas recessive are less. So dominant alleles are represented with uppercase letters, and our lowercase ones represent our recessive genes. So below shows the genotype for each. Stem cell or stem length is represented by two lowercase l's, and the flower color is represented by a capital F and a lowercase f. So, and we're going to be using these to do our Punnett squares. Keep in mind the recessive and dominant genes. So, we're going to watch this quick video.
Thanks for stopping by. Today we're talking about the basics of Punnett squares. Punnett squares measure the probability or likelihood of a genetic outcome based on a specific genetic cross. It doesn't tell you what will be, only what could be. For the simplest of Punnett squares, you'll cross two genotypes. These parent genotypes will be for the same gene but may or may not contain different alleles. Let's look at an example of a genetic cross for eye color. Green eyes being dominant and blue eyes being recessive. We'll use big G, little g for the maternal genotype and little g for the paternal. So this is our cross. The mother has green eyes and the father has blue eyes. And we want to see the probability that their offspring will have either green or blue eyes. For this cross, we'll draw a square split into 4 quarters, like a simple 4-panel window. This is the Punnett square. One of the parental genotypes will go on top and the other will go on the side. It doesn't matter which genotype goes where, and the order of the letters really doesn't matter, but we conventionally write the dominant allele first, if there is a dominant allele. So your setup should look like this. Then you simply carry the letters through to their respective quadrants and match them up, like so. Right? Each quadrant will represent a 25% chance that that genotype will result in the offspring. So in this case, we have only 2 possible genotypes, big G, little g, and little g, and each genotype has a 50% chance of showing up in the offspring. Similarly, you have 2 possible phenotypes, green eyes and blue eyes, with a 50% chance of each. Let's look at another example. We'll cross 2 genes that are heterozygous for eye color in this example. So big G crossed with big G, little g. These colors are just for illustration purposes. So we again will draw our Punnett square and put one gene on top and the other down the side, and then carry the letters through to their respective quadrants and analyze the results. So this cross results in a 1 to 2 to 1 genotype ratio, with 25% homozygous dominant, or big G, 50% heterozygous, or big G, and 25% homozygous recessive, or little g. The phenotypic ratio would be a 3 to 1 ratio, with 75% green eyes and 25% blue eyes. Okay, hopefully that helps you understand simple Punnett squares a little better. Genetic crosses can be...
So to explain this, when you use the word heterozygous and homozygous, I'm not expecting you to know or use those words, but hetero means more than one, so 2. And homo, that prefix means same. So this is a homozygous because they're both capital G. This is a hetero because it's a big G and a little g. This is also hetero, big G, little g. And this is homozygous, little g. We're looking at this. They got 25% GG. So both these are dominant traits. 50%, so two of these have a big G and a little g. And 25% have a little g and a little g. Keep in mind, when we talk about dominant traits, they are stronger than our recessive. So anything that has a capital G in it will be a green eye. So that's why they got 75%, because we count 1, 2, 3. 3 out of 4 is 75%. So 75% will have green eyes. Then if we look at our 25%, well, they got that from 1 out of these 4 squares has no capital G, no dominant trait, so it will be recessive, meaning 1 out of 4, 25%, will have blue eyes. Then we went through in the class, and we looked at our wonderful examples here. And this one, we're just going to be looking at the stem length, so how tall our plants are. So if we're looking at this, the lowercase l is recessive, which means it's a short one, and the uppercase L is dominant and means that one will be tall. So we look at our Punnett squares here. We'll focus in on this one. You have an L and an L, so a short genes, so a small plant, and a short gene, small plant, mixing together, combining, reproducing. So if we look, L, little l, go right over and up, so it's little l, and my computer will autocorrect the L's every time. But so we'll have an LL. Take this L, we carry it over here, and then we look up, you have another little L. So little l. We do the same thing here. We go over, there's a little L here, go up, little l. And the last one is the same, little l. So look at this. If we were to determine the percentage chance that this plant will be short, we can look through, we see all recessive genes, all little l. So 1/4, 2/4, 3/4, 4/4. So this plant will 100% be short. The opposite end of that, if we have two plants with 2 dominant chromosomes, so big L, and big L, and we breed them, what we would have is the same thing. Big L, look over again, is L over, look up, big L, carry it over and up, big L, and same thing, big L. And if we're talking about this again, the probability that this will be a tall plant, because it has all dominant traits, would be 100%. And the last one, this one was the tricky one. Tried to trick you guys. Worked for some people, not for all. So we have 2 plants we're mixing. One with they both have a dominant and a recessive gene. So they have a big L and a little l. So the way we go through this is the same way. Look here, carry that L in, look up. So we'll have big L and another big L. Look at this one. We have a big L and a little l. Same thing. We have a small L and a big L. We carried over, we'd have a small L first, then a big L. But remember, we want to make sure we put the dominant one first. So we have big L, little l. And then our last one, over little l. And then little l again. And then we talked about what would it mean if we're looking at the probability this plant will be tall. Well, we have big L for sure. Big L, little l. Again, remember, the dominant trait will overpower the recessive. The dominant's stronger. So these two will actually also be tall, even though there's a small L. They will be tall. So we have 1 tall, 2 tall, 3 tall. And if we think about that, 3/4 is 75%. So there's a 75% chance this plant will be tall. Then if we look at our 2 little L's, those are the recessive, meaning it would be short. So it's a 75% chance it will be tall and a 25% chance that it will be small. So that's what we talked about today in class. Your homework is these vocab words. So we want to make sure we get these vocab words put in. I'm going to go ahead and take a screenshot, and I'll put this as the thumbnail. So that way you know exactly what you need to put in your binder. If you have any questions, feel free to stop by the live session. I'd love to hear from you all. If not, have a wonderful day, and I will see you tomorrow. Bye.